All living cells are surrounded by a cell membrane, and the cell membrane has remarkable similarity from cell to cell to cell. The cell membrane, or what's sometimes called the plasma membrane, and those two terms mean the exact same thing, is composed of two primary components, lipid and protein. Now, this is a good opportunity for us to review what we call the 4 macromolecules. Every cell contains 4 macromolecules, large molecules that make up the bulk of its structure. Those include protein, carbohydrate, lipid, and nucleic acid. Now, some people want to substitute the word fat for the word lipid, and that's not correct. There are actually 3 different kinds of lipid in living cells, and fat is only one of them. So all fats are lipids, but there are some lipids that are not fats. And that includes phospholipids, the type of lipid that is one of the primary building blocks of cell membranes. When it comes to proteins in the cell membrane, there's a wide variety that we find. They have different functions. They have different jobs to do. You may be familiar, for example, with the idea that there are proteins in membranes that help transport things in and out of cells. But there are other proteins, for example, that function as receptors. Molecules that are outside of the cell bind to these protein receptors and cause things to happen inside of cells. There are some proteins embedded in the membrane that play the role of identifying a cell for what it is. And these cell identity markers, if you will, help the immune system in multicellular organisms know which cells belong in the body and which cells don't. And finally, when it comes to our microbes, some of the proteins that are embedded in the membrane make the microbe more or less virulent. In other words, when we're talking about pathogens, some pathogens are more deadly than others. They create a worse type of disease than others. And the molecules on that cell that allow them to be more or less pathogenic are called virulence factors. And proteins play a role in virulence factor structure. So proteins play a lot of different roles in a cell membrane, and they make up a big part of membrane structure. If you were to take a membrane off of any particular cell and bring it into the laboratory and split it into its component parts, about half of its structure would be made out of phospholipid, and about half would be made out of protein. Let's take a look at the phospholipids first. There's a lot of information on this slide. There's a lot of terminology that we need to become familiar with. We'll walk through it together. What you're looking at on the right-hand side of the slide is a drawing of an individual phospholipid. Now, this is a very simplistic drawing. They've included some of the actual chemical structure in the drawing, and then they've also just drawn very simple circles and straight lines and so on. But this is how we generally think of phospholipids, and it really is a pretty accurate depiction of how they're shaped. They have what we refer to as a head region, and that is sort of just a big sort of spherical glob on the molecule. And then they have these 2 independent, what we call tails, hanging off of that head structure. These are actually fatty acids. You may have heard of fatty acids in a previous course. The head isn't made out of one thing. It's actually made out of 2 different things. It's what's called a glycerol and a phosphate group. Glycerol is a small molecule that contains 3 individual carbons. Those carbons are attached to what are called hydroxy groups. 2 of them, at least, have hydroxy groups attached to them. The 3rd carbon is attached to the phosphate group. Now, phosphate groups are composed of a central phosphorus atom, that's the P, and 4 oxygens. Phosphate groups are very reactive. In other words, they're involved in a lot of chemical reactions inside of cells. Now, if we go down to the fatty acid tails, these are composed out of chains of carbon and hydrogen primarily. When we see a long chain of carbon and hydrogen on a molecule, we call that a hydrocarbon. When you look at this zigzagged blue line here, when you see that drawn, what the artist is trying to represent is a chain of carbon and hydrogen atoms. Everywhere there's a point on that zigzag line, that represents a carbon. And attached to these carbons are various amounts of hydrogen atoms. So here we have this long chain, this long hydrocarbon, and we have another one over here. Now you can see the artist has drawn this one very straight, and this one has a sort of a bend in it, a kink in it. The artist is showing us that sometimes fatty acids are sort of a linear in their structure, and those are what we call saturated fatty acids. That word saturated refers to the fact that there are no double bonds in this hydrocarbon chain. All of the zigzagged lines that are singular. There's one bond holding all those carbons together. Now, in the bent fatty acid, we see this. There are 2 blue lines here. That represents a double bond between 2 carbons. Whenever you see a double bond in a fatty acid, we now refer to that as an unsaturated fatty acid. Sometimes in a fatty acid there are multiple double bonds, and that is a polyunsaturated fatty acid. And again, you may have heard these terms in previous classes, particularly in a nutrition-related course. So saturated refers to single bonds. Unsaturated refers to double bonds. And when you have an unsaturated fatty acid in a phospholipid, you get this bend in the molecule. That's going to become important when we talk about how things cross membranes. All right, now the hydrocarbon portion of a fatty acid is only part of the molecule. So those 2 terms are not interchangeable. A fatty acid is composed of a hydrocarbon chain attached to this carbon with the double bond to the oxygen over here. This is called a carboxyl group. You can see there's a carboxyl group attached to both of these hydrocarbon chains. That's what makes a fatty acid a fatty acid. It's a hydrocarbon chain attached to a carboxyl group at one end. Carboxyl groups are reactive. They're going to be involved in chemical reactions. And in this case, they have formed a bond with the hydroxyl groups. Are you staying with me? Hydroxyl groups of the glycerol molecule. So the glycerol has hydroxyl groups coming off of it, and those are going to bind with the carboxyl groups of the fatty acids. And what do we get? We get one molecule that came together between the head, the glycerol and the phosphate, and the two fatty acids. Now, why do we care so much about this structure? Why do we talk in so much detail about the head and the tails? Well, it turns out that these two pieces of a phospholipid have very different chemical characteristics, and those are critically important to our understanding of cell membranes and how cell membranes work. So here comes a little bit more terminology for us. The heads in phospholipids have a property that we refer to as being hydrophilic. In comparison, the tails have a property we refer to as hydrophobic. Now, if you translate these terms, hydrophilic means water loving. Hydrophobic means water fearing. So hydrophilic things like to be near water. They like to associate with water molecules. Hydrophobic things don't. Hydrophobic things like to be away from water. All right, so here we have one molecule that has two very different properties. Part of it likes to be near water, part of it doesn't. Now there's some other terms in here. I've put the word polar in here for the heads, non-polar for the tails. We're going to talk about this in more detail in a later lecture, but polar is another word I want you to associate with hydrophilic. Polar things like to be near water. Non-polar things don't. They want to be away from water. All right. Now, the other thing I want to mention is this term, ester linkage. The way that the two fatty acid tails with their carboxyl groups attach to the glycerol molecule is through what we call an ester linkage. All an ester linkage is a covalent bond. It's a special type of covalent bond. And if you remember from your chemistry classes, covalent bonds are strong, right? They hold molecules together. All of these bonds down here in the fatty acids, all of these bonds up here holding the glycerol together, everything holding the phosphate group together, these are all covalent bonds. So an ester linkage is just a special type of covalent bond. It's what joins fatty acids to a glycerol molecule. Now, when you put a group of phospholipids together, and remember this drawing represents one phospholipid. When you put a whole bunch of phospholipids together in a watery environment, what happens is those molecules will spontaneously form a bilayer. Bilayer just means two layers. And our membranes, all cell membranes, are made out of these bilayers of phospholipids. Now, the bilayer has a very particular anatomy. The heads of all the phospholipids will be pointing towards the outside of the bilayer, and the tails of all the phospholipids will be pointed towards the inside of the bilayer. And that's because of this difference in their chemistry. Let's take a look at this structure that forms spontaneously in nature. This is a drawing of a membrane bilayer, or what you might read called a phospholipid bilayer. These are all individual phospholipids. And they have come together, again, spontaneously. You don't have to do anything to make this happen. You just have to put a bunch of phospholipids in a watery environment, and they will come together, and they will do this. Notice how they've come together. We've got what represents the outside of a cell here and the inside of a cell here. So you have to imagine that the whole cell would take up much more room than what's on this slide. We're looking at just a little piece, a little section of a plasma membrane, a cell membrane. All of the heads have oriented themselves to be facing the water. Remember, life on planet Earth is based on water. All living things are watery. So inside all of our cells, outside our cells, in our tissues, it's water that we encounter. So the heads are facing the water. They're facing the water in the extracellular space. They're facing the water in the intracellular space. They are hydrophilic. They like water. Now look at the tails. What the tails have done is oriented themselves in a way that keeps them away from the water. They're tucked in the middle. So we've created, with these phospholipids, a cell membrane. We've created this structure that has a very unique chemistry. A portion of it likes to be near water, and a portion of it doesn't. A portion of it stays away from water. Here's an important thing to remember about the membrane. These phospholipids, individual phospholipids with a head and two tails, a head and two tails. These phospholipids are not bound to each other. They are not stuck together with covalent bonds. They are simply associating with each other. They are attracted to each other because of their unique chemistry. So forces are holding the heads together, not bonds. Forces are holding the tails together, not bonds. Now, why is that important? Because it allows this membrane to be very fluid, to be very flexible, to move, in fact, and not be a rigid structure.